Well, good day to everyone joining us, and welcome to today's X-Talks webinar. Today's talk is entitled Using Artificial Intelligence to Identify Clonally Derived Cell Lines. See the future today. My name is Ryan Muse, and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes, and this presentation includes a Q&A session with our speakers. Now, the webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit your questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel, which is on the right-hand side of your screen. And if you require any assistance along the way, you can contact me at any time by sending a message using this same chat panel. At this time, know that all participants are in listen-only mode, and please note that the event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Advanced Instruments, who developed the content for this presentation. Advanced Instruments is a global company offering a novel portfolio of analytical tools, including Osmotech, a robust line of osmometers to support bioprocessing and quality control, and Solentum, a portfolio of best-in-class imaging and single-cell deposition technologies for cell line development workflows and assurance of clonality for regulatory bodies. Now I'd like to introduce our speakers for today's event. Paul Butler has worked across various functions in the life science industry, including sales, field application support, and product commercialization, with experience across a range of technologies, including automation, imaging, cellular, multispectral, and in vivo, label and label-free detection technologies, and next-generation sequencing. Currently, he is working on the launch of several new Solentum products that will enable more consistency across the cell line development process. Ann Davidson has the position of lead computational biologist at Advanced Instruments based in the UK. She currently works within the R&D department at Advanced Instruments, looking for new areas within cell line development workflows where artificial intelligence could be applied. Since joining, she has been involved with the production, validation, and deployment of new neural network-based approaches for multiple CLD processes. And Camilla Dominighetti has the position of biology manager, cell line development at Advanced Instruments, based in the UK also. As part of the applications team, her role includes overseeing the validation and optimization of the Solentum portfolio of technologies that improve cell line development and cell therapy workflows. In addition, her role involves improving the use cases and applications of these technologies to support Advanced Instruments for customers. Now, before we begin and continue on here, we have a poll question for audience members. This should be appearing on everyone's screen at this time, and you can participate by selecting on any of the answers that you see in front of you and then clicking submit. The question that we're asking is, how long does it take you per well to perform the image analysis in order to decide if a well contained a single cell at day 0? Your answer options are less than 1.5 minutes, 1.5 to 2.5 minutes, or greater than 2.5 minutes. We'll give everyone some time to consider their answer, and then in a moment, we'll hear from our speakers. The question is asking again, how long does it take you per well to perform the image analysis in order to decide if a well contained a single cell at day 0? Looks like most of you have submitted your answer, so thank you very much for participating. Let's take a look at where your results have come in. We have 44% for between 1.5 to 2.5 minutes, 36% at more than 2.5 minutes, and then 20% of you selecting less than 1.5 minutes. So thank you again for participating. Without further ado, though, I'd like to go ahead and hand the mic over to our first speaker for today, Paul Butler. Paul, you may begin when you're ready.
Thank you, Ryan, for moderating this session, and hello to all of the audience watching today. My name is Paul Butler, and I'm a Senior Product Manager for Advanced Instruments, focused on the Solentum cell line development portfolio. An important part of the cell line development process is the location and classification of cells within a microwell. It's a very time-consuming and labour-intensive process, and it's one that artificial intelligence is very suited to helping with. With that in mind, the goal of our project was to develop an artificial intelligence neural network that could help scientists to locate and classify cells. The idea was that it would do some of the heavy lifting involved, but it would still leave the final decision on clonality to the scientist. Anna Davidson, our lead computational biologist, will describe some of the factors that are involved in identifying cells, things like size and morphology, etc., and also how the artificial intelligence network was trained. Camilla, our applications biology manager, will then go on to talk about having developed the artificial intelligence neural network to explain how it actually performed in the real world when it was validated with real cell line development samples. And then at the end of the webinar, we're going to announce a significant new product. So what is artificial intelligence? The concept has been around for a long time, and in fact, in this 1950 paper by Alan Turing, the concept of artificial intelligence is implied, and he suggests a game called the imitation game, where a human is tasked with identifying whether another entity is human or machine. A more practical definition more recently is from IBM, and it talks about a field that combines computer science and robust data sets to enable problem solving, and also creating an expert system to make predictions or classifications based on input data. This is a nice definition because, as you'll see when we hand over to Anna, the artificial intelligence neural network that's been developed that we're going to talk about today was trained using robust data sets of images and has been trained to make classifications of cell types based on the input images. The use of artificial intelligence in cell line development isn't new for advanced instruments and the Celentum portfolio. In this case, you can see the VIPS, or the Verified In Situ Plate Seeding instrument. You can see in this image that after a droplet, hopefully containing a single cell, has been dispensed into the microwell, the droplet is imaged with a succession of Z-stack images. Finally, the artificial intelligence neural network will process the image and decide whether a cell was detected within that droplet. An artificial intelligence neural network is also employed by the ICON, which is used for productivity and titer measurements at the other end of the cell line development process. This application note outlines the validation of the ICON for cell counting and viability, and on this particular page you'll see reference to the fact that the assay is powered by artificial intelligence. So what we're going to talk about today is a new feature called automated evidence of clonality, and to understand what this does and its value, I'm first of all going to set the scene by going through the steps that are involved today for most scientists in cell line development in locating and classifying cells and making decisions on whether a cell line is clonal. So for most scientists, this is what the process looks like today. So on day 0, the day of seeding, you'll use either limiting dilution or an automated seeder, for example, to dispense cells into a microplate and then scan the plate using a clonal imager, and over the coming days, colonies will grow in some wells. Around day 14, or it could be day 7, or it could be later than day 14, you'll look at the plate and decide which of the wells do not contain colonies and those are then excluded. For the wells that have not been excluded, you'll begin to look at the images that were taken by the clonal imager, and typically you would start from later days in the process, where you can find the colonies and then begin to work back. In this case, you might start with day 14. Your next image might be day 7, but because you're looking at colonies, it's slightly easier to locate those cells. And eventually you'll get to day 0, and then you can make a determination as to whether a cell is clonal or not. The trouble is that locating the cells as you get close to day 0 can be tricky. It can take time, and there's also a lot of user bias involved potentially. Anyway, on day 14, after you've gone through this working backwards process, you can then annotate the cells that you judge to be clonal. And this process typically takes between 2 and 4 minutes per well, depending on the experience level of the operator. So having described the process today, I'm now going to hand over to Anna Davidson, and Anna is going to explain how the artificial intelligence neural network was created and trained to bring about the automated evidence of clonality feature. Over to you, Anna.
Thanks, Paul. So, as Paul has alluded to, the next addition to the studious artificial intelligence feature set is automated evidence of clonality. So, anybody who has done clone detection in day 0 whole well images knows the joys of searching around the whole well just to find that tiny little cell. So, to help with this customer burden, we have developed this AEC feature to help speed up this manual review. So, the cell metric has made its name for having incredibly even whole well imaging, with users able to clearly see to single cell levels. With this new feature, we have taken this one step further. So with an optimized imaging routine, we are able to get whole well images that have even more detail in them than previously. So having such good imaging has kind of opened up the door for having some equally as powerful image analysis. So with automated evidence of clonality, we have powered this by AI. So we have trained a neural network that is able to look at these high quality images and be able to detect cells and clumps at day 0. So as you can imagine, this is an incredibly powerful tool for speeding up that clone selection. This new feature will flag up single cells in addition to clumps and non-cell items for manual review. This non-cell category helps users identify those objects that kind of do look very similar to cells, but have been categorized as debris, so allowing the user to have all objects that are interesting immediately available to be scrutinized. So one fantastic description of this network was coined by one of our AI engineers, and that this network can be described as kind of automated Where's Wally or Where's Waldo. So it's immediately helping you to find Wally and confirm that they're there. So the next kind of thing I'm going to talk to you about is just to give you an overview of how AEC was developed. So the first step was seeding example plates and scanning them using Studious. So what we're then left with is a huge data set containing thousands of image examples with single cells, clumps, and debris in whole well images. Having such a large data set means we have many examples of all different types of cells. So cells that hide at the edge of the well look different to those in the centre of the well, for example, and we need multiple examples of each. What then happens is that each object in all the images were labelled up with their contents. So all single cells were labelled up, all clumps, and so on. The next step in development is picking and developing the type of network we want to work with, and what we landed on was an incredibly powerful, state-of-the-art object detection network that is up to the very tough job of single cell detection in whole well images. So once we have selected the type of network, the next challenge is training that network. This is where we leave the network with thousands of images and their labels. So the network will learn the characteristics of each class. So for example, it will learn that cells have a specific size, a specific roundness, a specific colour. We leave the network training for a considerable chunk of time, and throughout this process, we continuously test it on images that we know the contents of. So we can kind of continuously assess that performance. This process is repeated with continuous optimization of the network components until we get high levels of correctly labeled wells. Once the network has reached a point where we think that it has learned all it can from the dataset, we stop the training and proceed to a full validation. So, as Paul said, we currently have an extensive feature set that already utilizes AI. This process isn't new to us. So, we have many applications that support customers through that CLD workflow. Starting initially with the cell detection features within the Studios platform that powers the BITS Pro, we have an incredibly powerful network that gives an incredibly fast and accurate assessment of well content, so you can maximize your chances of getting clonal wells. Once your clone has been deposited in your well, that is where AEC comes in. So we are helping with that manual review of that 2-hour or day 0 scan. Once you have your clone, the next thing you want is an accurate depiction of that cell growth. So in multiple of the Solentum portfolio instruments, we now have an incredibly precise self-confluence feature. This is again powered by artificial intelligence. The neural network behind our confluence feature has been trained only to detect cell mass, and therefore any incorrect detection of the well edge has been removed. Once your clone has grown, if you're a CHO customer, the next hurdle is finding that ideal clone from the hundreds that you have to choose from. So we need to have an accurate count of the viable cell density for tighter normalization, and that's what brought about the development of the ICONS Trypon Blue assay, the foundation of which is an incredibly powerful neural network that has learnt the differences between live and dead cells, allowing for automated labeling, removing any lengthy thresholding procedures or manual counting. So all 4 of these networks roughly do a very similar job. They are finding those cells. And what makes these really special is that they require no customer input to set up. They have been trained so much that they know what cells look like. They don't need to be told that they're a specific shape in any capacity. It is really taking away that tedious setup and ultimately speeding up your manual review of that day 0 image. So now over to Camilla to talk about how this new AEC feature really does help.
Thanks, Anna. So now that you know how the feature works and why it was developed, let's have a look at automated evidence of clonality on some samples taken from the applications lab at Advanced Instruments. So firstly, I wanted to show you how easy and simple it is to set up the automated evidence of clonality scan. So here you can see the Studio screen, and this shows the simple steps that you need to follow for the preparation of your scan. You firstly select the project and the chosen plate, followed by the scan and the wells you wish to image. You would then simply place the plate into the instrument and select scan. So you can see from these steps how simple and easy the process is, and how there is no need for you to set up the focus through any adjustment or gating. So here is an example screen of a 96R plate where suspension CHO cells were single cell seeded, incubated for approximately 2 hours, and then imaged using automated evidence of clonality. So when you first click into the well, you are provided with a high quality whole well image that contains coloured boxes. And on the left hand side, you can see what these boxes represent. The green represents single cell locations, red cell aggregates, and white for non-cell locations. So you can see in this particular image that we have a single cell location highlighted close to the well edge. And if I zoom in a little bit closer, you have the opportunity to manually review and assess this object to see whether or not you in fact believe it is a cell. In this image, we also have a non-cell location highlighted by the white box. And again, you have the opportunity to manually assess this object and highlight whether you believe it is a cell or not. From here, you would continue to review the well and assess and highlight any other objects that you require. So one of the kind of major bottlenecks in this process is when you have objects that are hard to distinguish or identify. These can include cells that are close to the well edge, that maybe are a bit more translucent, or those wells where you need to look at their morphology in more detail. And here you can use our feature called the multi-plane imaging. This allows you to look at the cell at 3 different focal planes. You also have the ability to look at plate artifacts, cell or media debris, and any other objects within the well using this function. This makes your decision process a lot easier. So from all of this information, you can pull it together to make your unified decision on whether you believe a well is clonal or not. And this can be incorporated into the Studious software platform. You simply select the clonal tag at the bottom of the screen, and this will be recorded for any of your report generating. So here is another example well that has been scanned using automated evidence of clonality. This well contains more than one single cell and is therefore non-clonal. This can be easily seen from the green boxes that are highlighted from automated evidence of clonality. And as part of this process, you can again zoom into these objects for the manual assessment to make sure that what the network is highlighting is in fact 2 single cells. You can use the multi-plane imaging function to look at the cell at 3 different focal planes to verify your decision. And you can see using automated evidence of clonality makes this decision process a lot quicker. You can therefore exclude this well at this time, and therefore this will not be used as part of any of your future analysis processes. So as many of you will know, the process for reviewing wells for evidence of clonality is a time-consuming and labour-intensive process, making it one of the major bottlenecks in CLD. Therefore, we thought it would be beneficial to see how long this process would typically take when you have the support from automated evidence of clonality. So as part of this assessment, we had multiple scientists review a 96-well plate containing single-seeded CHO cells. As part of the assessment, they timed how long it took to assess these wells for evidence of clonality. The data from this challenge showed that it took between 30 to 40 seconds on average per well, and this was dependent on the user and their experience within this process. When you expand this time up to a typical 10-plate project, you can see the analysis taking between 8 to 10 hours with the support of automated evidence of clonality. And this is significantly quicker than the current timings using the traditional method for the manual assessment. As Paul mentioned earlier, it takes on average 2 to 4 minutes using the current method. For a 10-plate project, that can be between 32 to 64 hours, depending on the user and their experience. From this assessment alone, and the timings that we have shown, it indicates how automated evidence of clonality can significantly save you time in your manual review and consequently reduce your timelines to allow you to make decisions sooner in the CLD process. So now that you have seen how automated evidence of clonality speeds up your manual review, I also wanted to show you how you can utilize Studious further to speed up and support your CLD processes. Using the data generated from the automated evidence of clonality feature and from your manual tagging, you can use the triage function to highlight which wells to select or expand for the next stage of the CLD workflow. The triaging generates a list which can be exported containing critical data such as wells which have been tagged and confirmed as clonal, and even to include their percentage confluency at a specific time point, such as day 10 or day 14. Studious also allows you to generate a clonality report. This incorporates all of your crucial information, including those high-quality whole well images from your day 0 scan. This report can be used to support your regulatory submission. So I hope this has given you a good overview of how automated evidence of clonality can be applied in the CLD workflow to speed up your manual review process. And with that, I will hand back over to Paul.
So thank you, Camilla. And now you've heard from Anna how the artificial intelligence neural network was designed and trained using thousands of cellular images. It was then validated by Camilla using real-world images from cell line development laboratories. And now I'm going to talk about how the automated evidence of clonality feature has been incorporated into a brand new product called the Cellmetric X, and I'm announcing the launch of the Cellmetric X today, allowing you to do clonal assurance at day 0. So in terms of the new features of the Cellmetric X, we've already heard about the automated evidence of clonality, which allows you to see and assess clonality at day 0. The Cellmetric X is now integrated into the Studious data management and analysis environment, and this gives us the complete ecosystem of VIPS Pro for cell seeding, Cellmetric X for clonal imaging and confluence analysis, and the Icon productivity analyzer. We've also applied artificial intelligence to the confluence scan to give us a highly accurate confluence precision scan. And we have access to 3 planes of image data to help in the identification of cells. So before we talk about what the automated evidence of clonality feature looks like on the Cellmetric X, here's a reminder of the process for identifying single cells today, which typically takes 2 to 4 minutes per well, is fairly labour intensive as well. So now we can see the process for classifying clonally derived cells using automated evidence of clonality. So as before, we would seed the cells on day 0 and scan with the Cellmetric X. The difference, however, is that on day 0, we can now get the images, and in this case, automated evidence of clonality has suggested that this is a cell, and it's put a green box around it, which very easily allows us to locate that cell position. And then we can either accept the suggestion of automated evidence of clonality, or we can reject it based on our experience as a cell line development scientist. And the key point here is that automated evidence of clonality is supporting the scientist's judgment. It's doing the heavy lifting and the location of things that might be cells, and then allowing you to decide. I wanted to draw your attention to this example, and this shows basically 3 things that have been defined as cells in the well. The interesting thing is that for the cell in the top half of the well, you can see that it's almost on the point of cell division. it's still one cell, but it's about to divide, and the automated evidence of clonality has defined it as one cell. In the lower half, we see two separate cells in a doublet, and the AEC algorithm has picked it up as two separate cells and put green boxes around both of them. And this just shows the power of the AEC neural network. So now that the Cellmetric X has been incorporated into the Studious data management and analysis platform, we now have the complete Studious ecosystem, because the VIP Pro single cell seeder and the Icon productivity analyzer and the Cellmetric X clonal imager are now all operating under Studious. So that means that for the first time, there's a single data management and analysis platform that handles all of the data from seeding to final clone selection. This also delivers a fully automated clonality report and allows samples to be tracked all the way from seeding to final selection and master cell banking. Also, this allows us to triage samples. So because all of the data is held in one place, at the click of a button, we can triage samples based on, for example, clonality from the Cellmetric X, or single cell droplet data from the VIPS Pro. We can also triage based on the confluence level. So, for example, select only wells that have a confluence above, say, 70% at day 14. Or indeed, we can also triage based on titer and productivity, and we can have all of these different factors included simultaneously. Continuing with the theme of artificial intelligence, we have launched a new confluence precision scan on the Cellmetric X, which allows us to assess the confluence to within 5% of the reality in the well. In this case, on the left, we can see an iPSC colony. The green area is where the neural network and the human assessment agree, and the almost insignificant blue edge is where there's disagreement. Also for the HEC colony, you can see the blue shading being disagreement and the green area being agreement. In the case of the iPSC colony, this allows us to very accurately gauge the optimum time point for expansion without any risk of loss of pluripotency or other negative factors when deciding when to expand. The final new feature that I wanted to mention on the Cellmetric X is the ability to look at 3 planes of image data, and by that I mean for every cell that's imaged, you get 3 Z stacks, one taken from the close to the bottom of the cell, one from the middle of the cell, and one towards the top of the cell. So what you can see here is that we have a slider position which can be in one of 3 positions. In the middle position, you see that the green box around the cell shows the cell looking like that. If we now take the bottom slice, you can see that the image of the cell changes considerably. And if you look at the top, again, there's a clear change. When you're looking at cells, when you look at the 3 planes, generally you will see quite a distinct change in what those individual images look like. For something like a scratch on the plate or a piece of debris, there tends to be much less difference between those images, and this gives you a clue as to what is indeed a cell. So the Cellmetric X still offers the same high quality bright field imaging, the same level of contrast and resolution as was offered previously on the Cellmetric, Additionally, we still offer the 10-plate stacker option on the Cellmetric X, as we did on the Cellmetric, which allows you to load up 10 plates and perform walk-away automated imaging. And finally, we also offer a robotic access version of the Cellmetric X that can be integrated into high throughput robotic platforms via the flexible API and a barcode reader.
Well, thank you very much for this insightful presentation. I'd like to invite our audience to continue to send their questions or comments for the Q&A session. Before we get into that, though, we've got another poll question for everybody. This should be appearing on everyone's screen at this time. As with the previous one, you can select on any of the options you see in front of you and then click submit in order to participate. The question that we're asking is, what technology do you use to determine clonality currently? Your answer options are Cellmetric, other clonal imager, microscopy, or other. Give everyone some time to consider their answer for this, and then in a moment we'll hear from our speakers to answer your questions. Please do keep sending those in. The question, though, right now that we're asking of you for this poll question is, what technology do you use to determine clonality currently? Like most of you have your answer in, so thank you so much. Let's take a look at our results here. We have 48% of you selecting Cellmetric, 24% for other clonal imager, 12% at microscopy, and then 16% for other. So thank you so much for participating. We've got one more poll question here. This one's a simple one for you, perhaps. The question here is, for those in the audience using fluorescence labeled cell lines for clonality assessment, are you using GFP, or staining techniques, for example, cell tracker? So please select either of those two options, and then in a moment we'll hear from our speakers. The question again is, for those of you in the audience right now using fluorescence labeled cell lines for clonality assessment, are you using either GFP or staining techniques, for example, other cell tracker? Looks like most of you have your answers in. Thank you so much. Looking at the results here, we have 54% of you for GFP and then 46% for staining techniques. So again, thank you so much for participating. Now let's get into our Q&A session. I have our speakers here. Our very first question for you would just simply like to know, what if AEC is wrong? Paul, Anna, Camilla,
Yep.
perhaps you might
Yeah,
have some
Ryan.
thoughts.
Hi, Ryan. Yeah, thanks for that. Great question. So first of all, the purpose of AEC is to help the end user to make the final decision based on their experience. It's there to perform, if you like, the heavy lifting in terms of image analysis. And by that I mean, you saw that from Camilla's earlier presentation, that typically the user spends somewhere between 2 and 4 minutes analysing images to make a determination of clonality. And I think our polling answers reflected that number quite nicely. The idea of automated evidence of clonality is that things like locating cells, which is part of the analysis process, are done automatically. But we always expect that the end decision is made by the user. You know, if, and it can happen, if the automated evidence of clonality feature determines, for example, it classifies a cell as a piece of debris or the other way around, that sort of thing can happen, but we would expect that the user is going to make the final determination with the help of automated evidence of clonality. Great question.
Wonderful. Thank you very much for that great answer. I've got another question here, wondering, does the neural network continue to learn after the install?
I will take that one. Another great question. So this network is it doesn't continue to learn. So once we've trained it up, so we think it's learned everything it can from the data set that we give it, we stop that training. So we have a fully validated network that doesn't continue to go off and do unexplored things during your customer data set.
Very good to know. Thank you so much. So our next question here is asking, is AEC available on VIPS Pro?
So currently, the VIPS Pro is our automated single seeding system, and it's currently not available, but watch this space.
Very good. Thank you. Another question I have here coming in. Would like to know, would you be able to let the audience member know just how much longer it takes for imaging the 3 planes on the cell metric X?
Maybe I can take this one. So the 3-plane multi-imaging is actually included in the automated evidence of clonality scan and also in your Confluence precision scan. So it doesn't take any extra time.
Very good. Thank you so much. Next here, someone's asking, how long does it take to scan a plate?
I'll take this one as well. So for a 96-well plate, it takes around 12 minutes to scan for automated evidence of clonality.
Very good. Wonderful. Thank you. How about this question here? Someone saying that they saw examples shown were 96 well plates. They want to confirm that the AEC works for 384 well plates and the Confluency Precision works up to 6 well plates.
So it does work for 384 well plates. Part of the validation included the AI applications team running data through Corning 384 well plates. And in regards to 6 well plates, maybe Paul can comment on those. We are looking at working on 6 well plates, I believe.
Yeah, so what Camilla said is correct. Watch this space. So currently, the Cell Metric X is able to image 96 and 384 well plates. We will be releasing the other sort of lower density plate formats on the Cell Metric X in the coming months.
That's great. Wonderful. Thank you so much for those answers, all of the answers today. However, we have reached the end of our time here today. Now, if we couldn't attend to your questions, though, the team at Advanced Instruments will follow up with you. Otherwise, I want to thank everyone for participating in today's webinar. You will be receiving a follow-up email from X-Talks with access to the recorded archive for this event. A survey window will be popping up on your screen as you exit, and your participation is appreciated as it will help us to improve our webinars. I've also sent you a link in the chat box. And with this link, you'll be able to view the recording of this event on this page. And you can also share this link with your colleagues so they can watch it when they register for the recording here as well. So I encourage you to do that. Now, please join me once more in thanking our speakers for their wonderful time here today. We hope that you all found the webinar informative. Have a great day, everyone, and thank you for coming.
Thank you.
Thank you very
you.
much. Thanks.